Hello, in this video we will discuss about the Rash signaling pathway with Ip3 interact. So how to stimulate the Ip3 pathway we will discuss. So the both pathway is very important and crucial for the responsibility of the cancer causing cell. So the cancer progression is due to the Rash and Ip3 signaling pathway, but it is also used for the mitosis. So the Rash signaling pathway is involved in numerous cellular function, including cell proliferation, apoptosis also, but migration, fate specification, and differentiation. So this also is very important for the mitosis, and it is also important for the uncontrolled cell division. But when the but when the effector growth factor here is the person which contain tissue cell, so the effector growth factors EGF are present in the blood circulatory system. Where it's come from, we will discuss also. That will be bound with the tyrosine kinase receptor EGFR and to proliferate. And growth hormone, it can be growth hormone, insulin, IGF-1, IGF-2, and mitogen. So the mitogen is very important for responsibility about the mitosis and meiosis. So here is important thing is that this is the cell which contain a receptor. This receptor is the EGFR receptor. EGF receptor and plasma membrane and cytoplasm and nucleus. So when bind this mitogen, after the binding here is the DNA which that is activates the gene by the cascade of phosphorylation to become a transcriptional factor to use for the growth, mitosis and proliferation and differentiation also. So let's begin to understand first of all the endocrinology about that and how it's produced. For example, here is the hypothalamus which that is stimulated by the negative feedback, and the hypothalamus uncontrolled feedback can be. And in this way the adenohypophysis will release the somatostatin, and IGF-1 and IGF-2 is directly proportional to the somatostatin. And in this way important thing is that this is the important for cell growth and muscle growth, everything, and IGF-1 and IGF-2 also. So in this way the growth hormone will be released for growth. So the overproduction of the growth hormone will lead to cause the disorder as well as cancer. But in this case, we should not need to understand because the damage of the DNA is very important and crucial for the cancer causing agent. But in this way here, the growth hormone which that is our production will lead to cause the gigantism. So in this way here you can see this is also the pancreas is endocrine hormone produce somatostatin and insulin which that is also anabolic. So whole this story about the anabolism of the metabolism, so not catabolism. So the catabolism will be inhibited but the anabolism will be promoted during the eating time. So the fat state will promote these things, but in this case of fasting state will inhibit the proliferation and apoptosis will be occur. But during the eating time the apoptosis will be inhibited by the mTor pathway. We will also understand about that. So let's begin to understand and here. So here the EGF, the effector growth factor, for example, this is also known as ligand sometime, will bind with the EGF receptor, tyrosine kinase receptor. This is tyrosine kinase receptor. After the binding with the tyrosine kinase receptor, the phosphorylation will be occur. This phosphorylation through phosphorylase enzyme. So the phosphorylase and kinase enzyme. In this way, important thing is that after the phosphorylation, the EGFR will recruit the other protein from the cytosolic protein. For example, SHC protein, which that is phosphorylase and bind with the tyrosine kinase receptor intracellular domain, not extracellular domain, and the growth receptor binding protein 2. This growth receptor binding protein 2 will be bind again by the recruitment of the SHC complex, SHC protein. So after this GERB2 which that is recruit the source after the phosphorylation of the source To trigger the RAS, inactive GDP containing RAS will activate and dimerization with the GTP, and this activation with the dimerization with the RAS to RAF binding, and this is known as the dimerization. And this is the phosphorylation cascade which that is promote further, and in this way the RAF and RAF will be dimerization after the MAP kinase mitogen activated protein kinase, which that basically here is kinase will be activate the one kinase is used for the mitogen activated kinase 1 and 2 will be activated after the phosphorylation. After the phosphorylation through phosphate group donation from the MAP kinase to become a MAP kinase, and this MAP kinase will be more donate the phosphate group with the ARC 1 and 2, and ARC 1 and 2 is responsible by the triggering of the mitogen activated kinase 1 and 2, which that is basically activate the ARC 1 and 2 after the inactivation to inactivate the cytosolic protein will be activated, and this kinase protein will become a transcription factor. Here is the nucleus, so this go into the nuclear pore to enter, and the arc 1 and 2 will become a transcription factor arc 1, and in this way the SRF and SRF two domains bind with the ARC1, and in this way the helicase enzyme and polymerase enzyme will trigger by the activation of the gene and gene transcription to cell proliferation. After this, messenger RNA will be produced and go into the cytoplasm through a nuclear pore, and this messenger RNA which is very specific from the specific gene will use after the translational protein and anabolism will be occur. So this is the mechanism for the cell division and cell growth, as well as cell proliferation, cell differentiation, and inhibit the apoptosis also. So that is why the cell death will be delay, but the cell proliferation will be promote, and in this way the cancer causing cell will be proliferate due to the apoptosis. Rather than apoptosis, it will proliferate and cause cancer. But in this case, the if the ARC1 and 2 will be inhibit, so the messenger RNA will not produce and translation protein anabolism will be inhibit. But in this way, here is the basically the MAP kinase will inhibit the apoptosis. So remember about that. Apoptosis is very important for the inhibition of the cancer cell. So the repairing of the DNA is very important, but in this case the DNA will not repair, and the damaged DNA will proliferate with the cell during mitosis. So on the other hand, here is another mechanism we will discuss, Ip3 pathway interaction. So here is the phosphoinositide 3 kinase, which that is bind after the recruitment, and this bind with the RAS activated RAS GTP containing will trigger the basically here is the Ip1 phosphoinositide diphosphate, a bisphosphate, which that is present in the cytoplasm, cell membrane, and this will be phospholipase C enzyme used to cut this. But in this case, important thing you should need to understand, phospholipase enzyme C is not occur here, but you should understand. But here is the Ip3 phosphoinositide triphosphate will be formed after the phosphorylation, and in this way the PTEN or P10 is a phosphatase tensine homologue will inhibit this reaction. But in this case here the Ip3 will be removed after the protein kinase C will be bind and recruitment. How it's produced we will discuss, and diacylglycerol will be formed. And in this way diacylglycerol we will discuss here is the mechanism. So this is the phosphoinositide 4,5 bisphosphate is here, which that is basically is the Ip2. It is inactive, will be activated by a Ip3 formation, and in this way here you can see this is the this is basically the phospholipase C will be used for, and phosphoinositide 3 kinase is used for the phosphorylation. After the phosphorylation to become a phosphoinositide triphosphate, and in this way triphosphate is basically here. And after this, phospholipase C is used for the cutting of the diacylglycerol as well as the Ip3. So the Ip3 will be released and diacylglycerol will be released, and it is known as the DAG. So it will be released and protein kinase C will be recruited. How it's possible? Let's begin to understand. It is very important, and that is the important pathway is the Ip3, which that will recruit the protein kinase C. So here is the binding with the endoplasmic reticulum. So the endoplasmic reticulum contains calcium, and in this way inside the calcium is efflux by the binding of Ip3 due to the Ip3 sensitive calcium channel, and efflux of the calcium will trigger the cytosolic protein kinase C inactive will be activated, and in this way the activation of the protein kinase C will be bind with the diacylglycerol, and in this way the diacylglycerol binding the phosphoinositide Ip3 is also bind, but in this case the Ip3 is basically is the phosphorylation, and in this way the Ip3 is, you should need to understand the we will discuss later, but here is the phospho protein kinase C will trigger the phosphorylation of the other things to promote the apoptosis also, but it is also inhibit the apoptosis, and cell division, cell growth and proliferation, differentiation will be promoted. So in this way here the Ip3 is also used for the another pathway. In this way the phosphoinositide dependent kinase. So the phosphoinositide dependent kinase is trigger the phosphorylation of the AKT and PKB. This AKT and PKB will trigger the mammalian target of rapamycin mTOR pathway. In this way mTOR can be inhibited by the rapamycin drug, and this drug inhibit this mTOR pathway. But in this case you should not think about that, just you should think mTOR pathway is very important and crucial for the proliferation, cell growth as well as apoptosis inhibition. So the cancer causing mechanism is come from here. So remember about that. That is all thing about the Rash signaling pathway with Ip3 interaction as well as the mTOR activation, and on the other hand the ARC1 and the RAF and RAS activation. So thanks for watching. Please make sure to subscribe, like and share. Bye.